We report tensorial tomographic Fourier ptychograph, tofu square, a non-scanning label-free tomographic microscopy method for simultaneous imaging of quantitative phase and anisotropic specimen information in 3 dimensions. Built upon Fourier ptychograph, a quantitative phase imaging technique, tofu square additionally highlights the vectorial nature of light. The imaging setup consists of a standard microscope equipped with an LED matrix, a polarization generator, and a polarization-sensitive camera. Permittivity tensors of anisotropic samples are computationally recovered from polarized intensity measurements across 3 dimensions. We demonstrate tofu square's efficiency through volumetric reconstructions of refractive index, birefringence, and orientation for various calibration samples, as well as tissue samples from muscle fibers, or reconstructions of healthy muscle fibers reveal their 3D fine filament structures with consistent orientation. Additionally, we demonstrate reconstructions of a hard tissue sample that carries important polarization information for detecting cardiac amyloidosis. For more information, please check out our paper, Tensorial Tomographic Fourier Ptychograph with applications to muscle tissue imaging.